This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode number 218, recorded on May 28th, 2020. I'm Vincent Racaniello, and you're listening to the... Which podcast am I recording?
This is TWiM.
I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello, everybody. I hope all listeners are well.
I do, too. Are you well, Elio? You're still fine?
I'm fine.
Have you had any tests they run on you out there?
No, actually, the residence where I live, senior residence, does not have a single case.
That's great.
That's amazing.
Yeah, it is. They're very careful.
Also joining us from Ann Arbor, Michigan, Michele Swanson.
Hello. I am still on my feet.
And the weather's getting better out there, right?
It is. We've got a couple days of rain and thunderstorms as a cold front moves in. But yeah, it's pleasant. It's very green. Things are in bloom.
Also joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone. If it wasn't bad enough that we had COVID, yesterday we had our first tropical depression come ashore.
How was it? It was a surprise.
We had 3 inches of rain, a little bit of wind.
Oh my God!
But it rained like a Dickens, and then it blew itself out, and that's probably why Michele is getting a little weather today. Probably blew itself all the way to Michigan.
We have a lot of clouds today. Michael, are you guys ramping up there?
Yeah, we are indeed ramping up, but unfortunately, what we learned is that we saw our percentage double from last week's incidence of new cases in the greater Charleston area. We only had a 0.6% increase in new cases last week, and this week it doubled to 1.6%. So over the weekend, they had opened up a lot of social distancing, and people began to ignore their wearing of masks, and people hung out at the beach and weren't following the social distancing rules. And what we're seeing is it's reflected in the stats of new cases. I don't think it's due to the fact that testing is being more available, because we had plenty of testing in the previous weeks. So I think it may be our bad behavior has come home to roost.
It's a little early, isn't it? Well, I guess if people started the holiday on Friday and today's Thursday, they could already be showing symptoms.
No, we started back 2 weeks earlier.
Oh,
I mean, they
okay.
started partying on May 18th. And they have been going. So I think what we're seeing is the consequence of the governor's opening up the restaurants and everything else.
I see.
Early.
Not just the university.
No, not just the university. The whole city sort of let its guard down.
Oh, boy.
Michael, do you have plans for students coming back in the fall yet? Are they talking about that?
They are indeed talking about it, because I'm at a freestanding academic health sciences center, and most of what we do is experiential. I mean, we have dental students who work on patients. We have our 3rd and 4th year medical students who do rotations in our hospitals. We have nursing students. We have a large number of the allied health sciences that actually have practical experiences that you cannot simulate with Zoom, because you need direct patient interaction in order to accomplish it. So we are carefully looking at it. We're studying every aspect of masks and whatnot, and we're trying to look at things to try to understand what we can do to enable the best experience possible for our students.
Michele, you guys opening in the fall, do you know?
They haven't yet decided. They're still doing the analysis. And it seems like a long way off yet. I feel like we just got through graduation and finals.
We did, didn't we?
We did. That was last week.
Yeah, it was. All right, let's go on to our science for today. We're going to start with a snippet from Michael.
Well, today we're going to discuss a paper by Prather, Wang, and Schooley that appeared in the online edition of Science on the 27th of May, and it has the laudable title of Reducing Transmission of SARS-CoV-2. And this paper was literally brought to my attention late yesterday evening by Elio, and it caused me to scrap my plans to discuss an accelerated article preview that I was going to talk about that was in Nature that appeared a month before.
Sorry to lob you a curve.
No, that's okay, because, a month ago is a long time in the SARS world. And the paper that I was going to discuss was entitled The Aerodynamic Analysis of SARS-CoV-2 in 2 Wuhan Hospitals. And it was by an august group of authors, and that's available on Nature. And I'll put it in the show notes. It's worthy of your attention because they go through, and albeit they only looked at RNA, they did do a very careful analysis of how the virus is spread in the built environment of a hospital. But both of these papers complement each other. And much of the background information I looked at is germane to the discussion of today's paper, which is not science in the traditional sense. It's more perspective by these 3 authors. And it really frames the issues that we all need to be asking ourselves as microbiologists. And fundamentally, it goes back to, how did the SARS-CoV-2 virus spread so easily across our globe? I mean, we've only really been wrestling with this issue in the United States since March. 2 short months, March and April, and now we're through May. So 3 months, and we've literally seen a transformation in our culture.
But wait, the virus came before that, didn't it?
Oh, no, but in the United States, we've only been wrestling with it for 3 months. And so as of this recording today, our planet has confirmed the SARS-CoV-2 virus has infected about 5.7 million of the human race. And within the United States, this tragically accounts for 30% of that 5.7 million. So, we have about 1.7 million confirmed cases in the U.S. And unfortunately, we account for 28% of the deaths attributed to SARS-CoV-2, despite our advanced second-to-none health delivery system. And as yesterday...
Wait a minute. You're saying it tongue-in-cheek, aren't you?
Well, that is the point of this perspective. Yesterday, we passed the frightening statistic of the 100K milestone, something that a technologically advanced country should be asking, what could we have done differently? And I think any student of microbiology or epidemiology would offer one of the prime reasons that this virus has spread so easily is due to the fact that the human race has no herd immunity, with the replication number, or R naught, appearing to be about 5.7, with many of the infected being asymptomatic. And there lies the rub and the take-home message of this paper, which fundamentally tells us that mass and testing are a must if we have any hope of dropping that R naught, or multiplication factor, from 5.7 to the needed value of less than 1, so we can stop the transmission of this virus. And I'll put the R naught paper which is going to appear in Emerging Infectious Diseases in July, but it's available already as a reviewed preprint. And
Michael,
so
I
I'll
have to
put
say
that.
that paper is a bit flawed. My epidemiology friends say that it's not done correctly. It's higher than the usual value of 2 to 3.
Mm-hmm.
And there are a number of reasons for that, which we don't need to get into, but I would take it with a grain of salt.
Okay. Well, that's again why I'm mentioning it, because we need to, discuss all of the aspects of the science, because the only way we correct these perceptions of bad data is through, discussion. And after all, that is one of the reasons for TWiM. So the piece
and it'll be a challenge with a snippet.
Yeah. So the piece in our snippet today is a perspective. And Elio and I offer it to you, to our listeners, for I think it's an appropriate time at the end of our spring semester, if you will, to reflect on how far we have come in our understanding of the pathobiology of this virus. And it's now time, as the rise over run in the logarithmic curves seems to be leveling off, that we step back and see what lessons we have learned so that we may employ them going forward to battle this pandemic. And as I already alluded to, the simple message from their perspective is that mass and antigen testing are necessary to combat the asymptomatic spread of this virus that we are now seeing and think is responsible for the global transmission. And in support of this first, or their take-home message, is the observation that we need to appreciate that respiratory infections occur principally through this transmission of viral-laden particles in a size range of between 5 and 10 micrometers or microns. And aerosols are often less than 5 micrometers. And now these particles, these aerosol particles that are less than 5 microns in size, are the ones that have an easy ability to effectively be inspired or literally inhaled into a naive or uninfected individual if you're in close proximity to them. More importantly, an asymptomatic but infected individual can actually, through just their normal act of breathing and expiration, can expel viral particles in a cloud during breathing, speaking, coughing. The particles get a little bit bigger, sneezing. And in point of fact, we've all sneezed and hit our laptop screen or our computer screen, and the droplet size can get up to 1,000 microns in size. So humans can actually routinely produce respiratory droplets in size ranges from the ultra small, or 0.1 micrometers, which coincidentally can literally transition into the bloodstream, to as large as 1,000 microns. So here is where you need to remember what it means to be microbial, and that there exists a competition between the size of the droplet, inertia, gravity, and evaporation that in turn influences how far an emitted droplet from an infected person, either whether they be symptomatic or asymptomatic, can travel in air. And it's the big deal about how big these particles are. And the only figure affiliated with this paper is a really interesting one, in which they share with us the aerosolization, if you will, of how these particles move. And you literally see the infected and asymptomatic, and they offer how masks can actually limit and control not only the particle size that is emitted from an infected individual, but as well as the person wearing the mask, even though they're not a particulate mask that can exclude 95 to 100% of a particle of a fixed size, it really greatly reduces the risk associated with acquiring the virus. The other piece that we don't yet have a good handle on is we don't yet know the conversion factor between the cycle threshold value that we get from the PCR test, how many copies of the messenger RNA targets that the PCR has amplified. We have no way of determining the concentration of that, or it hasn't yet been published, the concentration of that RNA and how it relates to infectious particles. And secondly, we don't yet know how many infectious particles of the SARS-CoV-2 virus it will take to infect a susceptible individual. Have you seen anything yet, Vincent?
No, not at all. Very hard experiment to do in people, of course.
Well, ethically, it's impossible.
So we'll have to wait for animal studies to do that, but then you never know if it applies to people. So yeah, it's very hard.
So there are a couple of rules of thumb that you should keep in the back of your minds as you begin to think about it. The bigger the droplet, the faster it falls. And as it falls, you have to think about evaporation. And here, drier climates, such as deserts of California, Arizona, Nevada, Utah, are at an advantage. But we still need to remember that in our homes, our offices, our labs, they are often conditioned spaces. So they are intentionally not overly dry, and they often have a set humidity point of about 40%. So,
but Michael, at high humidity, the aerosol droplets that we spew collide with other water droplets in the air and then coalesce and fall more quickly to the ground. So high humidity is also safer.
And that is why Charleston is such a delightful place, because literally as we move into June, we literally can see the air that we breathe, or we feel the air that we breathe, simply because of the amount of moisture it holds. After Tropical Storm Bertha came through yesterday, you could go out and you could physically feel the humidity hit your face as you went outside to get a breath of fresh air. It was just laden with moisture. But what Michele brings up is that as these particles fall out of the air, this brings up the need and the importance of surface decontamination and cleaning, and why it's important to use one of these EPA registered disinfectants, not only in surfaces like we find in hospitals where there are a large number of viruses being expelled, but at the same time in our laboratories, where even when we're running shift work to effectively protect our postdocs, our staff, our students, we need to make certain that the surfaces that our students or our colleagues are encountering are indeed viral free. And again, gravity will help with larger droplets, but these smaller aerosols, this is in the size range less than 5 micrometers, will evaporate faster. They can settle, they're buoyant, and thus they're affected by air currents. So this then introduces the consequence and why we need to be wearing masks, because the mere act of just putting a barrier in front of your mouth as you speak can facilitate the knockdown of these smaller aerosols so that we don't expel them into the built environment where they can hang around for longer period of times. I'm looking forward to some of the studies that are likely being conducted now that are investigating the fluid dynamic models, looking at such places at football stadiums to learn if that will weigh in on whether or not there's going to be college football. Because in football stadiums, people are generally packed pretty densely. Even though they're outside, many would argue.
There's a lot of hollering and shouting.
Oh, there is indeed. And so I'm really looking forward to understanding those fluid dynamics to see whether or not we will indeed have college football this fall.
Well, you could play the game without an audience, right?
They can indeed. They can indeed. And again, the players could be tested. They could be PCR'd, or if they've already seroconverted, that would offer another opportunity. So we know. Now we got to put our microbiologist hat back on. We know there are two ways the virus can effectively infect us. It's either by contact, either direct or indirect. And the indirect method is where you touch a contaminated surface and then interact with your face and other mucous membranes, or this airborne inhalation. And it's really the airborne inhalation that I think is responsible. And initially, we didn't think it was responsible for much of the infection and why. And the data that the authors share with us is that when the nation of Taiwan instituted their control mechanisms, they actually did not shut down their economy. They did not even invoke social distancing per se. They just required their general population to wear a mask, and they instituted a process very early in the evolution of this pandemic in January, when, like every place else, they had very few cases, and they invoked contact tracing. And that result enabled the nation of Taiwan to have very few cases. And one of the interesting elements that these authors bring up in their discussion or their perspective is Taiwan has a population of about 24 million. And similarly, New York State has a population of about 20 million. Yet the data are dramatically different. New York did not invoke early masking and contact tracing when it had very few cases like Taiwan. And the consequences are quite different in terms of the number of infected and the number of dead that we see in Taiwan versus New York State.
Michael, can I ask you,
please?
Both the CDC and WHO, until not too long ago, did not recommend the use of face masks. They finally changed their minds, but it was too late. I cannot understand why. Do you have any insight?
No, and the perspective sort of touches on this. They point out that the 6-foot rule that many of us are so well familiar with is really data from the 1930s. And we all read with great interest the April 14th piece that in the New York Times that was based on a fluid dynamic model developed out of data generated at MIT that recommended 6 to 8 meters was really the safe distance in order to allow gravity, the humidity that Michele mentioned, to facilitate the dropping of the particles to effectively limit the risk. These authors are now advocating what Vincent just alluded to, the importance of putting masks on individuals, because very early on in the emergence of this pandemic, we did not believe, and this is largely due to bad data out of the early data affiliated with the German study out of Munich that said, well, the person was really sick, but they didn't tell anyone. And that's why, the folks in Germany got the virus. But what we now know is that very early on, the data speak to the fact that you can be infected in the upper respiratory tract and not be displaying any symptoms whatsoever, yet you're shedding large volumes of virus. And
I'm so curious about that. Vincent, are there other viruses that have that profile?
What, high shedding early in infection from the upper tract?
Before you're symptomatic.
Well, I think the other a good example is SARS-1, which did not do that, right? You mostly got very sick.
No,
quick.
and that's one of the interesting pieces that I hadn't appreciated is that the SARS-CoV-2 virus replicates 3 times faster in the upper respiratory tract than SARS-1 did. And
what about the flu viruses?
Other coronaviruses, they didn't comment on it.
Seasonal flu.
The flu, typically you have a day at the most of replication before symptom onset. So, very, very little, but much less asymptomatic transmission with influenza virus for sure.
Yeah, I think that's what's made this so difficult.
I think it's one of the main issues. Yes.
Yeah.
Yeah, and in fact, I think that's how it got to be a pandemic so quickly. Is that it went from an epidemic phase to a pandemic phase very quickly, simply because our past experience with viruses of the upper respiratory nature, we did not appreciate. And I think it's owing a lot to the fact that this virus replicates 3 times faster than SARS-CoV-1, and it can spread easily through the pharynx from before the innate immune response becomes activated to effectively make the infected individual feel unwell, or at least be able to forecast so that they can be a good reporter for contact tracing, which is another opportunity that technology is offering us. Many of you probably have read in the popular press that Google and Apple got together to activate the proximity sensors in our digital devices, our phones. And so that when we get close enough to each other, it will actually know that I have been close to Michele or Vincent or Elio, and it remembers their phone was near me. And then if you actually had an application that was developed by your public health department, it would then compare and contrast. Once I convert to being COVID positive, it would then alert everyone that I was in contact with within the last 14 days that I may have been exposed. So the bottom line of all of this is that you need to wear masks and we need to test. It'd be really terrific if we had a way of determining how many viral particles are indeed required to make us ill. And I'll put in the show notes the link to the New York Times virus transmission piece that they did in April, as well as some of the other things that we won't have an opportunity to discuss.
And also I have to put in a plug for This Week in Virology. The content there has been so fascinating. If those of you who want to really get deep into this multiple times a week, check out Vincent's other podcasts.
I'll have to check that out. Sounds good. Thank you. Thank you, Michele. Michael, we should wear face masks. We should be testing. The other day, the federal government said we're going to do 300,000 tests a day in the U.S. That's absurdly low. And Hospitals alone are doing 100,000 tests. I think it's a daily number. We have to do way more than that. And there's just this consistent unwillingness to admit that this is a problem and we need to deal with it.
And I can direct our listeners to many other podcasts that are going to take on what's going on in our federal government. Or not, as the case may be. Oh, my goodness.
In fact, to see this and to realize we should be testing extensively as Taiwan did. And
yes,
limit spread, they're a model for what to do.
Epidemiology 101.
All right, now we're going to do something very different. If you need a little break from COVID-19.
Yes, we do.
We're going to talk about a nidarian parasite of salmon that lacks a mitochondrial genome.
What?
It gets worse, Michele.
It is a PNAS paper from Tel Aviv University, Oregon State University, University of Kansas, the NIH, and the University of Montreal. The first author is Diana Yahalomi, and the last author is Dorothy Hutchins. And it's contributed by Ford Doolittle of Dalhousie University, is a well-known name in evolution. So, as we all know, we have mitochondria in our cell.
Powerhouse of the cell.
Powerhouse of the cell. And they make our energy by aerobic respiration. And we, of course, acquired the mitochondria many years ago during the evolution of eukaryotes. And most of the eukaryotes on the earth today have these mitochondria, and we can't survive without oxygen as a consequence. But apparently the loss of aerobic respiration has occurred independently in a number of eukaryotes that have adapted to low oxygen environments. Interesting idea there, right? You don't need oxygen, or there's none around, so you have to make alternative means, and then you lose your ability to utilize it because you don't need it, and it's a cost.
Use it or lose it.
Use it or lose it. That's right. Now,
evolution is mean that way.
When you lose a mitochondria, you have to have another way to get energy. Right, so you're losing oxidative phosphorylation pathways. You have to do anaerobic mechanisms. And that's what these organisms do that go into different environments. And they have what are called mitochondria-related organelles, or MROs, which they're no longer mitochondria. They don't have cristae, which are those folds on the walls of mitochondria, but they include hydrogenosomes and mitosomes. So this is a murky area. There's a little bit of debate in the field about what's going on, and that is what is this paper is about. They study a myxozoan parasite, a nidarian, which is a parasitic organism. It's a eukaryote. It lives in two different hosts, either in a fish and a worm, an annelid. And these are agriculturally very important. They can cause problems in fisheries and aquaculture. So they studied one of these myxozoans. This is the topic of the paper, and they find that it doesn't seem to have mitochondria. And there's pretty good evidence for it. And so, as you might guess, how do they do this? So we start with sequencing. They sequence the transcriptome, that is all the RNAs in this organism, and they sequence the genome, and they are comparing two closely related genomes, one that they believe has mitochondria and one that they believe does not. And so the one that, let me give you the two names here. We have Henneguia salminicola and Myxobolus squamalis, okay? And they're both parasites of fish. And the one, Henneguia and Myxobolus, Henneguia is going to turn out to not have the mitochondria, but they sequence both genomes and both transcriptomes. And so this is what they find. So Myxobolus, they're both related. Myxobolus, they can see the mitochondrial genome, which of course is a circular DNA in the mitochondria. It's a single chromosome. And You can tell by looking at the sequence that it's myxozoan. And they couldn't find this sequence in all the sequences they generated from Henneguya, H. salminicola. No mitochondrial DNA genome. And so you could say, well, maybe they missed it, but they did a lot of coverage of the genome. So what they did next was to say, okay, can we see it physically in the organism? So they have different developmental stages of both squamous and salminicola, and they stain it with DAPI, which is a dye that will stain nucleic acid. And what you would guess is that you're going to see staining of the nuclei, of course, because there's plenty of DNA in there. And in fact, both squamous and salminicola have nuclei. They better have nuclei. They're card-carrying eukaryotes, right? But the salminicola only stained in the nucleus and nowhere else. I should say that the squamous had nuclear staining and little dots in the cytosol, which of course are characteristic of mitochondria. Small blue dots in the cytosol, which are the mitochondria genomes staining with the DAPI, but no blue staining in the cytosol.
Diana said this was one of the amazing days in the lab where she opened up the photos that her co-first author, Stephen Atkinson, sent and was like, oh my gosh, you cannot. Yeah, it's just clear as a bell.
Did they do that before the DNA sequencing or after?
I think that they did the DNA sequencing first, and Diana wrote, she said it was amazing when they looked at she opened up the file and there were only 6 genes that were related to replication and translation of the mitochondrial genome. Yeah. So clearly it had lost a lot, thrown it out.
So here we have two pieces of evidence. No mitochondrial genome and no mitochondria staining in the cytoplasm. So they say, this is the sentence, this animal has lost its mitochondrial genome. Now, when you look at these organisms in the electron microscope, these Henneguya salminicola, the one with the salmon name in it, salminicola has organelles in the cytosol. They have cristae. Okay, so they look a little bit like mitochondria. They say they have a mitochondrial-like double membrane organelle. They have cristae. And, of course, in the other organism, they have cristae in their mitochondria, their normal mitochondria.
Are they called mitosomes?
Mitosome is a name for what this could be, right? The mitochondrial-like organelle, right? Yeah. Or hydrogenosome, where you're making energy via hydrogen. Now, these cristae of the mitochondria, if you remember your high school pictures of mitochondria, right? The wavy membrane, below the membrane, there's that thicker portion that's wavy. That's the cristae. There are genes involved in making that. And you can find those genes in both sequences of the two organisms, the squamous and the salminicola. So that makes sense because even though salminicola doesn't have a mitochondrial genome, it's got these cristae, and you need the genes to make them. So they're still present. So that's all consistent. No mitochondrial genome, but cristae genes, which are, of course, nuclear. That's where they would be. A lot of genes in the nucleus, by the way, encode mitochondrial components. So the mitochondrial genome only encodes a subset of what is in actually the mitochondria. Many of those genes apparently went into the nucleus many years ago and remain there. So most of what they say is most of the mitochondrial proteome is encoded in the nucleus of the cell. And so they can find many genes involved in key metabolic pathways. In both of them, they're still present in the nucleus. And so amino acid, carbohydrate, nucleotide metabolism genes, these are still in the nucleus. And these gene products would function in the mitochondrial-like organelle of these organisms. But almost all the nuclear encoded proteins, as Michele said earlier, involved in mitochondrial genome replication and translation were not found in the salminicola genome.
Listen, can I interrupt for a second?
Yeah.
We talk about loss of mitochondria. In theory, or at least in principle, there's another way in which these guys could have evolved, namely, they never got a mitochondrion.
Because we don't know if they were ever there.
That's right.
I was thinking about that.
But an organism that they never acquired a mitochondrion doesn't necessarily lack in evolution. It could evolve into anything. So in theory, it's not likely. Okay, I'm not saying this is a likely story, but in theory it's possible.
Yeah, I wondered the same. But the organelles that are present, they have cristae, which is a mitochondrial feature, right?
What that just means that they have to be able to generate an electron gradient, and they increase their surface area to mass ratio by the invagination. Each time you invaginate, you're creating a double membrane, if you will, and a single membrane unit. And that's effectively how Mother Nature then cheats the energy budget so that you can actually maintain the chemical gradient. Don't forget Peter Mitchell and the two components of his famous equation. You had the change in charge as well as the change in the chemical concentration. And in the case of bacteria, it's protons. And so it was that change in protons, because every time an electron would go into the membrane, you would throw a proton across the gradient. And then as the electron decayed through the membrane transport system, that proton then had to cross a membrane barrier again to make your ATP. And the hydrogenome is really no different in its energy design in being able to separate it, and it still makes energy in the form of ATP. So it's all about the ATPase at the end, getting back to Elio's question or statement that it may never have needed a mitochondrion because it was never exposed to the electron acceptor molecular oxygen. It had plenty of electron acceptors to dump it into.
But if I could interject, part of their data that argues that it's the loss is they see lots of pseudo genes. That is, there are genes that in other species are functional mitochondrial genes, but here they're essentially deteriorating, accumulating lots of mutations. So that's not what you would expect if they never had a mitochondria to begin with.
That's exactly right. Yep.
And that's how you make the argument. That's how you make the argument.
In fact, they highlight a mitochondrial DNA polymerase subunit gene, which in Salmonella is a pseudogene. It has point mutations that make stop codons, right? So it's a mitochondrial gene for sure. It's on the mitochondrial, it's on the nuclear genome because the mitochondrial genome is not present, but it's needed to replicate the mitochondrial DNA. But it was there, and it was presumably there because it was a mitochondria originally, and so the gene is not even expressed as RNA. And so I think that's correct, that supports the idea that these had mitochondria, because why would you have a mitochondrial DNA polymerase without a mitochondria?
Yeah. But they've got a lifestyle now where they can scavenge what they need or reside in environments that are suitable for their replication. So they're just slowly, the mitochondria are slowly deteriorating. Yeah,
so they wanted to know now if the loss of the mitochondrial genome is accompanied by loss of aerobic respiration in Salmonella, right? Because that would be a prediction. And they looked for homologs of nuclear genes that encode components of the mitochondrial electron transport chain complex, and nuclear genes for only 7 of these mitochondrial proteins. There are about 100 of them. Only 7 of them are left in Salmonella. So they've lost most of these electron transport gene encoding genes, and in the other organism, in other mixozoans, more of them are present. So they seek very specific complexes. If you're familiar with the electron transport chain, there are complex 1 and 3 and 4, et cetera, that are interacting, say, with oxygen, and they find key complexes are missing from some.
And they graciously include schematics that illustrate the chain reaction.
That's right.
To refresh your memory.
There's a nice picture. They show you what's missing.
Yep.
From Salmonella, and a lot's missing. So they say they probably can't do standard cellular aerobic respiration.
They can't breathe.
Right. Now, in contrast to the missing complexes, in addition to the missing complexes, most of the genes that encode a subunit of the ATP synthase complex, right, they're also missing. So that tells them a proton gradient is probably not present on the membrane, this inner organelle membrane in Salminicola. And in the other organism, the complex is present, so they can do aerobic respiration. So those are the data. They say that this Salminicola has lost its mitochondrial genome. It has an organelle that resembles a mitochondria, but this cannot use oxygen as an electron acceptor. So they would say this is a mitochondrial related organelle. It's not a true mitochondria. And by the way, Elio, they say these are not hydrogenosomes.
Okay.
They're distinct from hydrogenosomes based on some of the genes that are present and absent.
Let me ask you a question now. Are these the most evolved animals that are totally anaerobic, or are there others? It seems to me that I've heard in the back of my head that there are animals which are totally anaerobic. Therefore, they don't need a mitochondrion. Do you know anything about it?
Yeah. I don't know the answer to that. I mean, they talk about other protists without mitochondrial genomes.
Not protists, animals.
Yeah, I know. But they don't mention any of those. I would think that they would, right?
I would think so.
I was not familiar with these weird and wonderful creatures. I love the last sentence of their paper that states, as a highly diverse group with more than 2,400 species that inhabit marine, freshwater, and even terrestrial environments, evolutionary loss and simplification has clearly been a successful strategy for the Myxozoa, which shows that less is more.
That's great. You like that? Yeah.
Terrific.
So let me make one more statement about the habitat of this parasite. So I said it alternates between two hosts. In the fish host, it proliferates in white muscle, and this is a tissue known to have anaerobic metabolism. So that's consistent with it not having mitochondria. And then the other host is a worm or an annelid, and many of these members of this family grow and reproduce in anoxic environments. So we have the two places where this organism lives are low oxygen environments, and so maybe the loss of mitochondria was driven by that. And they mentioned that, in these small organisms, in these organisms with small genomes, the length of the genome, the nuclear genome of these is in millions of bases only. The cost of carrying an extra gene is bioenergetically very high. So they think it makes sense that these have been lost because they're not needed anymore. And so it all makes sense.
So I was able to communicate with both the two co-first authors, and they too have led really interesting paths to their careers. So I'd like to share their stories. Diana Yahalomi was born in Argentina. When she was 4, the family immigrated to Israel. And she said she was a shy girl, wasn't very good speaking Hebrew, but she learned how to use her sense of humor to manage embarrassing situations. And she relayed an example, which was when she was interviewing for the master's program at the University of Tel Aviv, a professor said, why didn't you get into this other prestigious program that you applied to? And she just shrugged and said, I guess I was a false negative.
Love it.
Isn't that great? So she did a master's with Tal Popka, and then on the recommendation of another professor who didn't have an opening. And in that lab, she fell in love with bioinformatics, but she also made great friends, including another Tal, who later introduced her to her husband, and they now are raising 2 amazing kids. But then after her PhD, she wanted to go from being a simple biologist to also being an expert as a computer scientist, because she thought later she'd just get a job in bioinformatics or programming. And that's when she joined this lab, Dorothy Hutchins' group, studying these fascinating creatures we heard about today. So she was productive in more ways than one. She was 5 months pregnant when she defended her thesis. Initially, she hadn't planned to stay on and get a PhD, but because in Israel, the academic life is really accommodating to working moms, that she said, even though it was hard to go to work after being up all night with the baby, she's really grateful that she had the opportunity to do that. So she got her 2 different bachelor's degrees, one in biology, one in computer science at Tel Aviv University, and then a master's also at Tel Aviv University and using computational approaches. And then her PhD was also at Tel Aviv University in the Department of Zoology, and that was the work that we heard about today. Currently, she is a bioinformatics analyst at the Weizmann Institute of Science in Israel in the Center for Personalized Medicine. So she would like to share that one of the most exciting things in life happened by chance. So she advises people, don't waste time feeling bad about what you might have missed or things that didn't go as planned. She said, that's just how life works. So she was a bioinformatics specialist on this team. The other co-first author is Stephen Atkinson. He's a senior research associate professor at Oregon State University in Jerry Bartholomew's research group. And he thinks that he's taken an unconventional path, probably because he's always followed his interests rather than having some kind of long-term concrete occupation. So Stephen is Australian. He earned his bachelor's at the University of Queensland, majoring in geology and chemistry, and then did a 1-year project on metal detection in water. But then Stephen stepped away from academia to follow another of his childhood passions, photography and graphics. So he actually co-founded Wildfire Studios, which is a computer gaming company that he did the graphics for, that created several computer pinball games, including Balls of Steel, which I personally haven't played, but maybe someone out there is familiar with it. So he stayed with that company for 6 years, but he maintained his interest in science, in part because his girlfriend, who's now his wife, was studying parasitology. So together they moved from Australia to Germany and then the U.S. for his postdocs. And he, all this time, was really captivated by discovering new species out in the field and then using the microscope to capture their images and then later incorporating the DNA sequence analysis to kind of gain insight. He is currently part of the faculty at Oregon State, and he is continuing to study these fascinating microscopic parasites. He says they're everywhere, any place that fish and worms reside, marine and freshwater. And of course, he's been able to do field work in the Pacific Northwest.
So much for sushi.
Yeah.
So much for fishing.
In the Pacific Northwest, of course, salmon fishing is a major industry, but he's also done some work in Michael's backyard in the Charleston Bay. So he'll go out in the field.
I appreciated that. I appreciate what you sent to me last night. That was cool.
And they've got their work is funded by the U.S. Department of Interior and also Oregon's Department of Fish and Wildlife. So he also, beyond his lab research, he's really committed to science outreach. So together with his supervisor, Jerry Bartholomew and Andrew Myers, they're faculty advisors for an interdisciplinary student club called Seminarium, where the students use cross-disciplinary approaches to create exhibitions on campus and for the general public. He's also organized workshops for the Peak Adventure Club, which is for kids K through 5th grade. And he created a website and is the webmaster for this public resource called fishpathogens.net, which includes a lot of educational images and content on these fish parasites. He also uses paint, paper collage, digital illustration, 3D printing, and his own scientific expertise to engage the wider community in science by creating art pieces, which he's shown in galleries and public spaces. Yeah, so his advice about science is that serendipity is ever present, and you just can't predict where a project or question will take you. So, keep an open mind and bring all your skills.
Nice. Thank you, Michele. I'm sorry also earlier when I started, I'd failed to mention the two co-first authors, Diana Yahalomi and Stephen Atkinson.
And, one situated in Israel, the other at Oregon State. So, yeah,
pretty far apart, which works. We know from these days now it works for sure. All right, so let me read an email here before we wrap up. This is from Sophia. Hello, TWiM professors. It was great to hear Dr. Schmidt on TWiV.
Oh.
Even though I didn't catch what he said, especially pertaining to the virus going to the heart, but this is the wrong podcast to ask this. Anyhow, this is a question for Elio. Happy belated birthday, by the way.
Oh, thank you.
And it has to do with fungal aerosol. So fungi can be around in the air. And here's what happens when you combine lockdown, heat, and humidity. And she sends a link to a story. Business reopens its doors after 53 days of closure due to quarantine, only to find all of the leather products moldy.
Ooh. The image is something to see.
Moldy shoes.
Oh, God, yes.
I wouldn't wear those.
No.
Oh, everything in the store is moldy. Purses, backpacks. Wow. Even the chairs you sit on to try the shoes on. So now, if I remember correctly, there was this story told by Vincent on TWiM about a research lab that had gotten flooded and there was mold on the walls, but the lab kept functioning until the lab members became sick with a respiratory fungal infection and some of them died. So how worried should we be about fungal aerosol? Let's say in the store on the link. Is it safe to clean the mold up without wearing any type of mask? And why, if I remember the flood story correctly, would a non-pathogenic mold become pathogenic or symbiotic to a dangerous degree in your lung? Another case of what repeated exposure to something can result in? I thought I would ask a wise man. That would be Elio. Thank you for your dedication.
Oh, my goodness. Thanks for the flattering remark. And boy, is it misplaced. I don't know. The answer is I don't know. However, I think it's a numbers game. The fungus does not become a pathogen overnight just because it's been around. But if you have a lot of humidity, you're going to have lots more fungal spores around. And fungal spores in large numbers are certainly not good for your lung. So I suspect it's simply a possibility that if you increase the moisture in your environment, you're going to have more fungus.
And a heavier load increase the risk.
Yes. I also think that if you're cleaning out the store, you should definitely wear masks, right?
Oh,
absolutely. Yeah. Masks will work very well to eliminate fungal spores. So if you're worried about it, a mask is the thing.
The other thing I understand is that in many people, many of us have fungal spores in our lungs, but only when you're immunosuppressed can that be a problem, right? So It could be that some of the people who got sick were, somewhat immunosuppressed to begin with. So
possible. Sure.
In
fact, it's likely.
Yeah. Another concern is all the stagnant water in hospitals and hotels that haven't been used.
Oh, the Legionella. The Legionella issues.
Legionella could be, if they're not properly flushed before people occupy those buildings, there likely be more cases of Legionnaire's disease.
And remember the trick, if you're concerned about a hotel room, is if your grocery store still uses plastic bags, get yourself 2-3 rubber bands, put the plastic bag around the shower stall, and then taking the pen that the hospital leaves on your nightstand, poke 3-4 holes into the bag, and you put the bag over the shower head, and you turn on the shower and allow it to run. It will fill up the bag with water and not aerosolize it, and a water stream will come down so you don't actually run the risk of inhaling the Legionella, and you just have a stream of water that can actually effectively flush the pipes if the hotel isn't hyperheating their water to inactivate the Legionella.
I think I'll stay home, Michael.
Well, you like to travel, Vincent. You're always jetting about, going to exotic locations.
This year, I haven't gone anywhere. And the funny thing is, last year I flew 75,000 miles. Okay?
Oh, my God.
Mm-hmm.
I got platinum status on United. I was so excited, and it's going to go to naught because this year I have 0 miles, and I won't use it, and it expires.
No, United extended it a year. You'll actually get it for the following year. All of the airlines extended it for the year.
Michele, so hotels typically have water tanks on the roof? Is that right?
I think there's such wide variety. I would hesitate to say, motels, hotels. Yeah.
And if they have it and it's been sitting, it could be growing Legionella. Is that the problem?
But there are ways they could flush it out before put it back online.
Got it. Well, here in New York City, we have lots of rooftop water tanks for sure, right? It's the way you get water.
People are home using those water tanks.
Yeah, they're home. That's true. That's good. The hotels are not.
Yeah.
They've probably been flushing more than normal.
Right.
Probably, yeah. All right, that's TWiM 218. You can find the show notes at microbe.tv/TWiM or asm.org/TWiM, either place, same notes. If you want to send us a question or a comment, TWiM@microbe.tv. If you like what we do, consider supporting us financially, microbe.tv/contribute. Not just TWiM, but all the other science podcasts that we do. Michele Swanson's at the University of Michigan. Thank you, Michele.
Thank you all, and stay safe.
Elio Schaechter is at the blog called Small Things Considered. Thank you, Elio.
My pleasure. Thank you.
Michael Schmidt at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank ASM for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.